All right, so it is my great pleasure to introduce Dr. Amber Van Laar. Yeah, come on up. She and I have known each other for a number of years. We worked at University of Pittsburgh together. She's a movement disorders trained neurologist who now is very involved with clinical trials and particularly clinical trials regarding the last two slides that I just threw up. So I'm going to let you take it from here and put a little bit more detail to that, but I think you're going to enjoy this. Thanks, Amber.
All right. Good morning. I think it's still morning. It's a real pleasure to be here. I've been looking forward to giving this talk for quite a while. Today, my focus is going to be on cell and gene therapies for Parkinson's. I am a company employee for AskBio, so I've got some biases. These are my disclosures. If you have any questions, let me know. So I'm going to start off with a lot of, actually kind of, I think what you may have heard this morning. Like there are still some unmet needs for Parkinson's. So really one of the big topics that we are trying to address, a lot of what I'll be talking about today is, what are some ways that we may be able to actually slow down or stop Parkinson's disease? But there are a lot of other issues. I'm sure many of you could add things to this list, but what are the other ways that we can stop the progression of the non-motor symptoms? These can show up very early on in the Parkinson's disease course, and yet there's a lot of therapies that do nothing to address this, and these continue to get worse over time. So is there a way for us to address those kinds of symptoms as well? And then another big issue is falling, freezing, other gait issues. The ability to walk and be mobile is critical. And yet some of our therapies can address some of these symptoms, but at the end of the day, there are still a lot of problems that people have with falls in particular. And then another big issue for a lot of patients, and many of you probably have felt this, but you take medicine and symptoms get better, but symptoms come back, and this is kind of constant up and down throughout the day with medicine. So what can we do to help get a more even keel across the day so that the good on symptoms can stay more for longer parts of your day? And what I want to really focus on today, as you just heard, I've been a movement disorder specialist. I've sat in clinics with patients for many years. And at some point, it gets really frustrating when I feel like I can't do anything to actually really address the underlying disease. And that's really what led to my interest in cell and gene therapies. And so today, I'm really just going to drill down on those two major topics. So why do I think that cell and gene therapies could address some of these unmet needs in PD? So what's different about cell and gene therapy? For many years, I think this can still sound sort of sci-fi to do cell and gene. And one of the reasons I think behind that is that we usually have to administer these things through surgery. There's a blood-brain barrier that I think you may know about. And because of that, there are a lot of times that we have to actually directly put the type of therapy into the brain itself. And so we'll talk about that a bit more today. But deep brain stimulation is another example of this, right? We have to kind of go around the barriers that Parkinson's put into place and put the electrodes right into the brain to get that benefit. But with cell and gene therapy, a lot of these clinical trials, if we're going to be doing brain surgery, there should be a big effect here. It can't be something that's modest. We need to really be able to feel it. And so a lot of the clinical trials right now are looking at how do we get that big effect, and then could we actually change the course of the disease? Maybe we actually be able to slow down or stop Parkinson's in its tracks. For everything I'm going to talk about today, I'm really trying to focus on what's available here in the United States and what's active right now, or what may be imminently coming in the next year or so. So just keep that in mind. Okay, we'll start with cell therapies. I think the best way to think about this is we're trying to replace those dopamine cells that are being lost. So for many people, I think cell therapy, you're replacing cells. It's intuitive. It makes sense. Those cells are dying. Put those cells back. If those are the cells that are dying, let's just replace them with more dopamine cells. And in some cases, some of the therapies you'll see, these are cells that have been modified, so they are basically just little pumps of dopamine. So you put them, into the brain, and you can just really pump out dopamine instead of constantly needing to take the pills. So these are a great idea. So where are we right now with these cell therapies? So I think the first way to think about this is replacing what? Where are we putting these cells? And so if you might know some neuroanatomy here, the substantia nigra is where the bodies of those dopamine cells live. So if you kind of follow that little blob there, that would be like the body of the cell, and then it sends projections up into the putamen. And the putamen is where a lot of the working end of the dopamine goes. When you have Parkinson's, there's less dopamine, so there's less movement of dopamine to the putamen, and that's what leads to, with less dopamine, that's the stiffness and slowness and tremor and difficulty moving around. So I'm pointing out the putamen here because a lot of cell and gene therapies will target that part of the brain. It's easier to get to, it's a bigger target, and that's really, again, where the business end of dopamine is happening. So let's start from the basics here. What are stem cells? I think there's a lot of ideas out there, but let's kind of start with some basics here. So think of stem cells like the raw material. So all cells come from a stem cell. We have many types of cells in the body, and these stem cells theoretically can turn into any type of cell. So an eye cell, a heart cell, a toe cell. In some occasions, we are able to take stem cells, and we can, in a lab, like in a dish, we can force them to turn into certain types of cells. And in the case for Parkinson's disease, we're looking at kind of pushing them to turn into dopamine neurons, so the brain cells that make dopamine. So I think there's a lot of places that we can get these stem cells, and this can be a hot topic and can be a little bit controversial at times. But there are a lot of places that we can get stem cells. So there's autografts. So this is taking cells from yourself, they're auto, versus an allograft. So this is getting cells from somebody else that you then put into another person's body. There are xenografts. These are taking cells from animals, and this has already been done for Parkinson's disease, believe it or not. There are also iPSC cells. So these are basically your lab-made cells. So you can take cells from an adult, convert them to a stem cell, and then tell that stem cell turn into a dopamine neuron. And all this happening in a dish. It sounds kind of sci-fi perhaps, but this is a really hot way of doing stem cell work right now. So there's a lot of iPSC work that you may see in the literature. Now, more controversial, and I think what may turn off a lot of people to stem cells in general, that we can get stem cells from embryos as well. And this has been another methodology that's been used already in treating Parkinson's disease in the past, just FYI. So again, we can take existing dopamine cells from something, like even an embryo, or we can reprogram cells. And a lot of efforts are going away from embryonic stem cells and using more of these lab-derived, like we're making these cells in the lab instead. But here we are. There are stem cell studies still going on right now, but they aren't active. You can't prescribe stem cell therapy. What's holding things up? So part of the problem here is, for a long time, we had no idea, how many cells do you put in? What's the right type of cell to use? Is one better than another? Okay, once we get them in the brain, how do we get them to talk to each other and talk to the parts of the brain that are still there? We have a network that's already been built, that's been established. In a lot of cases, we're putting into somebody who's 60, 70, 80 years old. Like, how do we get those cells to start talking? Is there a right person? Is there a right type of Parkinson's patient that we should be considering for cell therapy? And then on top of that, how do we get it into the brain? Like, how do we safely put cells into the brain that will work? And then the other big issue that is being faced with cell therapy is that there's immune suppression that's needed. So medicines like steroids and things to quiet the immune system while the body is trying to incorporate the new cells. So these are a lot of questions that the therapies that are out there right now, the studies that are ongoing, they're asking all these questions right now. So it's a slow, methodical process to help us understand what's really happening. So let me go into some of the studies that are going on right now. So as full disclosure, Blue Rock is part of our company, AskBio. So this is our cell therapy company. We work together with Bayer Therapeutics. There's been a lot of discussion and news about this company and their stem cells. So they were using an embryonic stem cell, and these were cells that were, again, being asked to turn into dopamine cells. So this was a phase 1 study. So this is early. This is open label, meaning that everybody knew they were getting treated. So there was no control arm. And this was a dose escalation study, meaning that they had higher and higher doses in different groups or cohorts of these patients. It's delivered by neurosurgery, and it's a one-time treatment. So this isn't something that you have to go back and do again. It's a one and done. Again, the cell type they used here was an embryonic-derived dopamine neuron. And so some of the upsides, the way that I kind of see this is that it's nice. You're taking those dopamine cells, you're putting them back into a dopamine cell deficient brain and asking them to make more dopamine. Now, with more dopamine, this should help with the symptoms that improve with Sinemet, with levodopa. So tremor, stiffness, slowness, these things that improve with levodopa should get better with these cells that are now making more dopamine. Now, some of the watch-outs, and this is going to be true for any of the cell therapies, are the cells going to survive? Do they make it through the transfer? Did they stay in there? Are they incorporating? These are big questions, and that's also really hard to show clinically. Like, how do I show the world that, yes, those cells are there and they're working? It's tough. Another watch out for cell therapies, and this is for all of them, again, there's immune suppression that's needed. So again, you have to take medicines after you've had this procedure. And just as an FYI, there's a phase 2 study that's being planned for this year. So just keep an eye out for this study if you're interested, and happy to answer any questions you might have. So, big question. It was a safety study. So was it safe? So actually, this just came out very recently. There's been some presentations at big Parkinson's meetings. And so no major safety issues. And I actually would say this has probably been one of the safer stem cell studies that have been done so far. Right now, it's still early. They've been able to watch these patients for up to 18 months. And we'll have to continue to watch them. But so far, so good, this is a big hurdle that many other studies didn't get past. Now, a big problem in other stem cell therapies has been that after they've transplanted those cells, some patients get really bad dyskinesias that they cannot control. They're called runaway dyskinesias. So this has been a big issue in the past. We think it may be because of how it was being delivered. But fortunately for this particular study, they didn't have any issues with this. So it's a major step ahead from some of the stem cell therapies that have been done before. This study also involved immune suppression. So they had patients on this for 12 months and then watched them for another 6 months without immune suppression. And it looks like they didn't have any issue with the immune suppression or when they came off. It wasn't like there were new symptoms that showed up. So this is, again, major advancement in our progress for cell therapy. So keep your eyes out. This is a study that's really up and coming. I mentioned this one here because this is one of the oldest groups. In Sweden has done a ton of stem cell therapies. And right now this is only open to participants in the UK and Sweden. They opened up an observational study so that if you wanted to get into the stem cell study, you had to be in their observational study. But this is also a phase 1 study, so meaning this is beginning of trying to understand their therapy. And it's open label, again, so everyone knows that they're getting treated. It's also neurosurgical delivery, going to that putamen, again, the sort of the business end of the dopamine cells. And I put this down here because I want you to appreciate, like, how many cells survive? Implant 3.5 million, they're only expecting 1 million of those cells to survive. And then the higher dose is 7.1 million, only expecting 2 million to survive. So this is, again, some of these questions we're asking ourselves, like, just because we get them in there, do they work and are they alive? So, one of the upsides to this is, again, very experienced groups. Some of the very earliest stem cell study was done in Sweden, so this is a group that really knows what they're doing. But again, this is limited to the UK and Sweden. This is one of their highest doses that they've ever delivered, so interesting to see how that goes. And as I said before, immune suppression is still needed for this one. This is another interesting study. So this is allogenic, meaning that, again, coming from somebody else, it's cells from someone else that you then put into your body. So that makes it a little bit different. They're using fat cells. So this is, I think everyone would like to get a little fat cells to get some dopamine cells out of it. So this is interesting also because it's a phase 2, meaning that they're going to have a blind. There are going to be some patients who are getting placebo. And so it's a placebo-controlled, and it's hard to know. You'll never not know which group you get into. So either you get these these adipose-derived mesenchymal cells, or you get saline. This is not a one and done. This is 7 infusion, 20 weeks. The study lasts a year. This is great because it's IV administration. You're using your own fat cells. Who wouldn't want that, right? But some of the watch-outs here, we right now there's no mention that if you are in that control group, are you going to then be available to have the active treatment? That is not clear. And if you're putting brain cells into your IV, how do we know they're getting into the brain? I think that's a big concern that I have. But maybe they do. So this is an ongoing study right now, I think mostly in Texas, but it's available here in the United States. So this is another autologous, meaning it's coming from self, so using self cells. This is not an active study right now, but it looks like it's imminently coming. So there's an upcoming phase 1/2 study. This is very interesting. This is, again, using those iPSCs, so these cells kind of forced in a dish to turn into dopamine cells. Very recently, the study actually just received fast track designation, meaning that they should be able to have, go a little bit faster through the FDA system. But just keep an eye out. This looks like it should be starting kind of soon. And happy to share these links with anyone. My other big watch out here is there's a lot of medical tourism with cell and gene therapies. There are a lot of studies being done, and if you start looking, you can find stem cell therapies for Parkinson's and many other things that you can just go to a local clinic. So just be very cautious when you're looking at these things. Please vet anything that you want to potentially participate in with your neurologist. There are a lot of things that are not done to the same rigor that we would expect for things happening here in the United States, so just be very cautious. Any study that you have to pay to get into, I would be very, very cautious about that. A real clinical study you shouldn't pay anything for. Okay. So again, what are the holdups? We've known about cell therapies. We've been implanting these things for decades now, so where are we right now? Why aren't we further along? One of the big issues is this manufacturing. So think about it. If they want to take my fat cells and turn them into dopamine cells, that process can take a long time. And then to do it with the rigor that we need to make sure it's clean and that it's not going to make you sick. There are very strict rules on this. It's really hard to manufacture those cells in a way that the agencies want to see. And we still, what is the best cell type? We really don't know. We've had a variety of cells that have been tried, but this question still remains. And then what's the best way to deliver them? This is also still a big, big question. They're fragile. You can't just shove them in. They have to be very gently deposited into the brain. And so this has been a hold-up in getting this more real time. There are still some ethical concerns. There's still embryonic cells out there, and this is still an issue for many people. The immune system suppression is still a big thing, but I would argue maybe not as big of a deal as we thought it once was. But this concern for those dyskinesias, so these kind of wild flailing movements, I think we feel a little more confident after this most recent study, but it's still watch out for all of the stem cells. Now, ultimately, the other problem here is we can get those cells in there, we can take a look at the patients and think that they're looking better, moving around better, but we still need to prove to FDA and other regulators that they're there and they're working and they're alive. And so how do you show that? And that actually is also a big unknown right now that a lot of other studies are trying to figure out. So I wish they were a little bit further along, because if you look at the survey, this was in 2019 for the Michael J. Fox Foundation. So I think they were actually surprised by this answer. But how would you consider having a stem cell therapy? And I ask yourselves, read these answers and ask yourself, how would you have answered this question? But I think what's really most compelling from the survey is that the vast majority of people said yes, maybe with some caveats, but vast majority said yes, they would consider doing a stem cell therapy. So again, this is a hot topic. I think this is a lot less sci-fi than it used to be. There's a lot of activity happening in this field right now. Okay, I'm going to switch over to gene therapies. This is more near and dear to my heart. I've been working on this in the lab and now in the clinic and trying to make this for reality for Parkinson's patients too. So I'm going to talk about gene therapy, but in order for me to kind of do that, I need to start at the very basics here. So we're going to go back to school, gene therapy 101. So with gene therapy, we're taking advantage of the normal mechanisms that every cell has in the body. You have genes. Your genes are your DNA. DNA is made up of all your genes. And when you have your genes, the big point here is that the genes get read by machinery inside your cells. Every cell does this. And they get turned into proteins. I think I had heard some discussion about this earlier today. So we have our gene that sits in our nucleus. It gets read, and then it makes proteins. And those proteins are the business end of our genes. They're the ones that do the work. They're also what give us the eye color that we have and the color of our skin and make our skin flexible. I mean, proteins do everything in our cells. And again, gene therapy can work in a variety of ways. So sometimes, we deliver these genes into cells, and those genes then get turned into proteins, and that's actually what works like the medicine for us. And so instead of taking a pill, we're using those genes and like our medication. And there's several ways that we can do this depending on what kind of disease it is that we're talking about. So one way is you introduce a healthy copy of a gene. That's really a lot of what I'll be showing you today. And so you ask the cells to make a good copy of a gene that you think may be helpful or beneficial to the dopamine cells. And in other conditions, you may want to turn off a bad gene, a gene that's making a protein that's toxic. But you can also replace a non-working gene or an unhealthy gene with a healthy copy. So there's lots of ways we can use gene therapy, but for today's purposes, really focusing on this first one here. So when we're talking about gene therapy, we're talking about trying to deliver genes into cells. The best way to do this is the best delivery packaging that we have is an envelope, and our envelope in this case that we're talking about today is AAV. So this is a virus that's been stripped down. It cannot make you sick. So all the components that would contribute to the virus replicating and making you sick, all that's been removed. AAV itself cannot make humans sick. So what we use is just the shell, just the outside of this virus to work like our envelope. We know viruses are good at getting into cells. That's what gives us colds and everything else. But in this case, we just take the outside to use to kind of capture our gene. So and then that's how we do the delivery. So if you think about our gene is like our letter, and that our capsid, our AAV, is like our envelope. We have to stuff the envelope and put our gene inside, the message inside our envelope. And then in our case, we have a brain surgery, so we deliver it right to the brains. This is like our delivery truck. And then when you have your full package and it gets next to the cell, it knows what to do. It delivers the DNA into the healthy cell. So in our case, we're delivering this to brain cells in people with Parkinson's. And then the cell does what it normally does. It reads the message and says, oh, okay, I need to make more of this protein, and then you get more of the protein being made in the cells that have received this copy of your gene. So this is really the bread and butter, the basics of how gene therapy works. So I'm going to a lot of attention here to, this is one of our studies from AskBio. This is looking at GDNF gene therapy. This is a compound that's been looked at for a long time for Parkinson's disease. Just to mention, so AskBio is the cell and gene therapy unit for Bayer Pharmaceutical. We're one of the leading platforms for gene therapy, been around for over 2 decades, working on AAV gene therapy. So we have a long history here, though you may not have heard of us before. I probably don't need to tell anybody in this room, when you have Parkinson's disease, that putamen just is not seeing as much dopamine because the cells are being lost because of Parkinson's disease. And so with less of the dopamine neurons, less dopamine's around, and that again is what leading to stiffness and slowness and tremor. Now, GDNF, so this is a growth factor or neurotrophic factor. It's food for brain cells. So GDNF's been looked at for a long time in Parkinson's disease. And we know that for those dopamine cells to even start to develop, you have to have GDNF. Without GDNF, they do not develop. In adulthood, so after you've been developed, you still need a little bit of GDNF to keep those cells healthy. And we actually also think that it's some of the loss of GDNF that may contribute to the onset of Parkinson's. So you can take down sort of the helpful GDNF, and that may be part of what allows Parkinson's to happen. The hope really here is that with GDNF gene therapy, you restore the health of these cells. You bring back this growth factor that's been missing, and you improve the health. And this will also allow us to improve the neurons and the brain cells that have been sick. They're not doing well because of Parkinson's, but they're still there. They haven't died yet. Can we bring those back to health, resuscitate those cells? And again, the hope here is that by doing it this way, that we're treating symptoms, but not just symptoms, that we may actually be treating the underlying disease simply by just restoring the health of those dopamine cells that have been injured by Parkinson's disease. So AV2-GDNF gene therapy, so this is our compound that we're using. So this is also delivered neurosurgically. This is a cartoon of how it's being done. So we take a one-time procedure in the back of the head. We deliver this gene therapy to the putamen, again, that kind of the brain center right kind of in the middle. It's a one-time procedure. This is done inside an MRI machine, so we can see exactly what we're doing in almost real time. So we have just, the MRI is just constantly going. And the advantage here is that we can customize and tailor these infusions to every, not even person, but every putamen and how that is shaped. So we put the medication just there. This is a technique that's been evolving very rapidly, but it's been going for over 15 years of being developed in order to be able to put drugs into the brain. This took a lot of advancement in technology just to get there, but I think we finally have that part down. And it's an advantage here, one time. This is not something you have to go back and do again. It's brain surgery, but it's a one and done. The other big benefit, especially over something like DBS, there's nothing left behind except the gene that we wanted to be there in the first place. The envelope, just like you would at home, if you've got an envelope, you package it up, you throw it away, and everything that's left behind is just your letter or your message. So there's no hardware, there's no batteries, there's nothing to manipulate like you would with DBS. Otherwise, your neurologist would just be managing things just like they normally would with medication. So GDNF, again, we think is really helpful in restoring the health of these dopamine cells. And again, we need those dopamine cells to be healthier, to function better. We do think this will help with the motor symptoms and keep those fluctuations, from happening. We also hope that, GDNF will stop those continued cell death, so we can kind of, again, stop it in its tracks. And we're watching right now, is this going to, how long can this effect last, and how much effect are we going to be seeing in these motor symptoms? And again, the real hope here is that this is not just fixing the symptoms, treating the symptoms, that we're actually having an impact on the underlying cause of Parkinson's disease on top of that. So again, these are really hard things to show to the FDA. So this is a video of one of our infusions from our phase 1B study. So what I'm going to show you here is a phase 1B. It's still going on right now, but we're not recruiting for it at this time. Again, this was open label, meaning that everybody knew what they were going to get. And this was, again, neurosurgically delivered, went to the putamen again, one-time treatment. The one advantage compared to a lot of other cell and gene therapies is we have a lot of experience with this particular drug. So this was looked at as far back as 2013. So we have a decade of experience of this AV2-GDNF gene therapy in people, which is rare. This is a huge advantage. So one of the upsides to GDNF gene therapy is this ability to restore the dopamine system. So we're taking the cells that are already there, trying to make them healthier and work better, and doing that to help relieve the dopamine deficient symptoms. Again, stiffness, tremor, and difficulty walking. And another upside is just a one-time procedure. And so, as you can imagine, this procedure takes a while. It's very, very carefully done, so it's a long procedure. GDNF's been looked at in a variety of ways, and in some cases it looks like maybe there was something there, but in other cases it just flat out failed to meet its primary endpoint. A lot of it, I think, was the problem with delivery, which I would argue we've now been able to overcome with this procedure. So let me just point out what's going on in this video here. So this is an MRI. So this is actually many, many MRIs that have been stitched together into a video. This procedure actually takes many hours. It's just been sped up for purposes for having a video here. So just to orient everybody, eyeballs are up here, ears will be here. This is the back of the head. So you're seeing this line here. So that's actually a cannula, or basically a needle that's going into the back of the head. There are two. So there's one here. There is another one on this side. It's just a little bit out of the plane. You can't see it. But what you're seeing is this cannula is going to the putamen. That's this comma-shaped structure, this grayish blob here. And what you're seeing is that that white is our gene therapies. That is our study drug. And we have a little bit of a contrast agent in there so we can see exactly what we're doing in real time. So you see a little bit of white start to form, and then once we fill up that structure, we advance that needle and fill that part up a little bit more, and then keep going until what we do is fill up that putamen and only the putamen. And we get to watch this, again, with the MRI going constantly so we can see, make sure that we know exactly where we are, we know exactly where it's not going, and we know when we filled up the structure and we're done. So there's some huge advantages to that. And if there is a problem, we have the MRI going the whole time we'll see if there is an issue. And there's a lot that we can do in the middle of the procedure if need be. So this has been 30 years of technology that took us to get here. But I think from a delivery standpoint, which has been a massive limitation for cell and gene forever, I think we finally have overcome this. There's a phase 2 study that's being planned for this year, opening shortly. So if there's any questions about that, my colleague Juan Ortiz is here as well. We have a table with some fun stress balls back there too. I'm happy to answer any more questions about this particular study. Okay, moving on. So again, a major question, this was a safety study. Was it safe? So we had no major safety issues with AAV2 GDNF gene therapy. We have more than 18 months data at this point, but the surgery was well tolerated. People are doing well. At our 18-month time point, when we're about to announce this to American Academy of Neurology in like a week and a half, we looked at patients with mild PD. So this would be about 2 years from diagnosis on Cinemet, but small doses. And what we found in this group is that they were their motor symptoms were very stable, that they looked like they were able to maintain now for quite a while. And I think for a lot of people very early on in their PD course, if they could just kind of stay there and not get worse, I think a lot of people would be happy with that. We need to continue to watch this group for, right now it's for a total of 5 years. So we're watching closely. If we can maintain, where people were at year 2 for 5+ years, I think that would be quite significant. But what's really interesting is what our moderate group. In our moderate group, we saw really significant changes in UPDRS scores. So that's the tapping and how fast you move. But beyond just the scores that our neurologists rated, the patients themselves too also noticed a big improvement in their symptoms. So stay tuned. We have a phase 2. It's also starting. Again, we're so impressed by what we found in the phase 1B that we're moving on to the phase 2. And again, happy to talk about that more. So there are not many other gene therapies going on in the United States right now. There are only 2 more I have to talk about. So the AAV-GAD, so this is one that was tried before. So this was already looked at, was kind of promising. I think the company pulled it back, kind of retooled a little bit, and are now putting it back out into the study you see here. So this is a randomized study, meaning there's some blinding here. They have a sham controlled surgery. And this is one exception. So they're putting this in the STN, the subthalamic nucleus, which is a different location because they're trying to, again, use this as a disease-modifying effort and trying to change the way that the brain is signaling in the dopamine system. So it's a different kind of approach here. There's some historical experience with this one, so I think that this isn't brand new, which is a good thing in that I think that they've improved their delivery technique, and this will be an interesting one to watch out for. So this is open to a lot of patients with Parkinson's. Some of the other studies are very restrictive. This one's open to pretty much everybody. And again, I think this is nice in that it's a little bit different than anything else that's out there. So just if you're interested in this one, it's a controlled surgery study, meaning that there's going to be people that have a sham surgery. And it's not known, again, if they will be able to be offered the active treatment after they've gone through the study. So these are things to watch out for. And one more. So this is for AAV-GBA. So this is for patients with a known mutation in this gene called GBA. This gives a kind of a different flavor of Parkinson's disease. It's nice to have something that's very specific. I think when people think gene therapy, it should be going after like a gene. And in this case, it is working that way. They do a different kind of delivery. So it's going into the cisterna magna. So that is this fluid-filled space kind of behind your ear. So they're not going into the brain at all. So it's a bit different kind of procedure. And this is looking for more advanced patients, so with more severe Parkinson's, but also you have to have that GBA mutation, so that limits who can get into this study. It's nice in that they're replacing an enzyme. They're replacing something that's missing, and so that is also kind of a unique mechanism here. And it's also open label, meaning that everybody's going to get treated. They also have immune suppression in this study. And again, it's good and bad, but only people with known GBA mutations are eligible for the study. Okay, so I've said a lot. Hopefully something sticks, even if there's just one idea. But really just be careful what and where you get your information. My first place is always go talk to your neurologist. They're going to be a wealth of information for you, and they know you, right? They know you, and they know your Parkinson's, and they know your family dynamics. So please always check with your neurologist. But the Michael J. Fox website, they have a Fox Trial Finder, which is fabulous because you can actually look at locations. It's easier to maneuver. They have, and the studies that are really dedicated to Parkinson's, they have them all on the Fox Trial Finder. There is a government website. I hope some people are aware of this, the clinicaltrials.gov. This is a great website. Any study is required to be listed on clinicaltrials.gov. So if there's something you're interested in and they do not have a listing on clinicaltrials.gov, it probably is not a legitimate study. And I will say it again, if you have to pay to get into any study, it's not a legitimate study. So please do your homework, talk to your neurologist, talk to your family. There are a lot of other research companies, us being one of them. We're happy to answer questions to the best of our ability. There's a lot of universities. If you look on the clinicaltrials.gov, you can see the locations of where these studies are being done, and you can contact those sites. They give you that information, so you can reach out right to the source. And then again, talk to your neurologist. They're a wealth of information. We have a table out front. There's happy to answer any questions. We also have Ask First at askbio.com. This is a great resource. We can sign you up for our newsletter if you're interested in kind of hearing where we are with Parkinson's and moving forward. So keep that in mind. That's really it. I will hang around if there's any questions. I also got done a few minutes early. But thank you for your attention. I've been really looking forward to giving this talk for many years. Thank you.